Dear students, welcome by Sri Chaitanya Academy for this live KCET 2025 biology paper discussion. Let us dive into the paper discussion right away. So coming to this question, it asks, with respect to the limitation of the ecological pyramids, which of the following statements are correct? So here, the first statement, it does not take into account the same species belonging to two or more trophic levels, is a correct statement. It assumes a simple food chain, something that almost never exists in nature, is a correct statement. It accommodates saprophytes, is the false statement. It does not accommodate a food web, is also the correct statement. So therefore, the correct answer for this question is going to be option number 2, A, B and D. Next question. This is from the chapter Biodiversity. The sixth extinction of species presently in progress is 100 to 1000 times faster than the previous 5 episodes of mass extinction. This is a direct question from the textbook. Species diversity. Now here you need to understand, when you move from equator to the tropics, the weather stabilizes, there is more availability of the nutrition. So the diversity is going to increase. Now as you are going from the tropics towards the temperate and then towards the poles, there is a steep change in the climate and the availability of the nutrition decreases. So therefore, species diversity decreases as we move from the equator to the poles. Therefore, for this question, option number 4 is going to be the correct answer. Coming to the next question. This is from Principles of Inheritance and Variance chapter. This is the pedigree chart. Now, let us call this as generation 1, generation 2 and generation 3. Now here we can clearly observe that males and females are equally affected. As a result, this is going to be a autosomal disorder. Now if you look at generation 1 and generation 2, nobody is affected, few are affected. Now if you look at generation 2 and generation 3, no one is affected, few are affected. So this is a characteristic of the recessive trait. So hence we will conclude that the pedigree chart under question is given for the disorder of the autosomal recessive type. A classic example would be that of sickle cell anemia. The next one, this is coming from Anatomy of Flowering Plants chapter. So here it is telling me that the vascular bundles are conjoined and collateral. The minute we see these two words, you understand that it is for a leaf or a stem. Then it says open. So when you combine both of them, directly we are going to arrive at dicot stem as the answer. Next. Now this is a question from Cell Cycle and Cell Division chapter. Now here you please pay attention to this word. Now in anaphase, the chromosomes, they have split the centromere and they are moving to the opposite poles. Now by the time we reach to the telophase, the chromosomes have completely reached to the opposite poles and they will lose their identity and they will become chromatin. Now since the question says it is to be placed at the opposite poles, for this question telophase is the most appropriate answer. Next one, identify the incorrect statement with respect to the rules of binomial nomenclature. This question comes from the very first chapter, the living world. Let's look at the options. Biological names are underlined separately when handwritten. This is correct. Biological names are printed in italics to indicate their non-Latin origin. So this is the incorrect statement, because when you italicize a word for binomial nomenclature, you indicate the Latin origin. So therefore, for this question, the incorrect statement is option number 2. Next one. This is coming from the chapter Biological Classification, Kingdom Monera, Classification of Bacteria. So here coccus would mean spherical, so A going to 3. Directly I can remove options 2 and 3. This is what we call elimination trick. Then bacillus is going to rod shaped, B going to 1. So directly I have the answer, that is option number 4. Next, read the given statement. So this question is coming from the chapter Plant Kingdom and it refers to bryophytes. So here it is telling me that gemmae are green unicellular sexual buds which develop in receptacles called gemma cups. Now here you have to pay attention, because gemmae are not uni but they are multicellular structures, and they are not sexual but they are asexual buds. So therefore, from this we understand statement 1 is incorrect. Now let us look at statement 2. Protonema develops directly from the spore. Now here they are referring to mosses. Excuse me. Protonema directly develops from the spore is the correct statement. So therefore, statement 1 is false and 2 is true. Therefore, option 1 is going to be the correct answer. Now, identify the types of aestivation in corolla labeled as A, B, C, D. Now, the minute you look at this, you know this is a characteristic aestivation in family Fabaceae, and this has to be vexillary. So, I can remove option number 4. Now, I can come here, this is the simplest one, and this is going to be valvate. So then I can remove option number 2. Now C is going to be twisted or contorted aestivation, where there is a particular direction in the arrangement of the petals. They not only touch each other but they also overlap in a particular order. Then this is going to be imbricate. So therefore option 1 is going to be the correct answer. Now here again we are presented with another question from Anatomy of Flowering Plants. So here they have given us characteristics of vascular bundle, and you need to identify its transverse section where we are going to observe it. The minute you see radial, it has to be root. Then you see tetrarch, it has to be dicot root. So A has to go to 2. A going to 2, I will remove options 2 and 3. Then second one, conjoined open and endarch. Now the minute you see endarch, it is for a stem. Then you see open has to be dicot stem. So B is going to go to option number 4. Directly I have my answer, that is option number 1. Next, the reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are the membrane free system that are going to float in the cytoplasm. Next, the cell wall-less prokaryote among the following is mycoplasma. This is a direct question from the textbook. Next, now here you are given with the prophase 1 of meiosis 1, the substages and the key events. We are required to match them. Now leptotene, we all know, here we are going to start seeing the chromosomes under light microscope. So A has to go to 2. So look at the niceness of this question. If you are able to get A is going to 2, you directly arrive at the answer of this question. Then let's proceed to the next question. Now this is a question coming from photosynthesis chapter, combining C3 and C4 cycles. The given statement, statement 1, in Calvin cycle, carboxylation is characterized by peptidase enzyme. False, because in a C3 plant, the carboxylation is always by the Rubisco enzyme. So therefore, statement 1 is going to be false. Now let us look at statement 2. In a hatch and slack pathway, that means it has to operate in the C4 plant. Here the carboxylation, we have two of them. One is the primary carboxylation, which happens in the mesophyll cells, that is by the pepcase enzyme, and the secondary carboxylation is going to happen in the bundle sheath cells and is catalyzed by the enzyme RUBP carboxylase. Therefore, statement 2 becomes correct. So hence, first option is going to be the correct answer. Now, this is a question from respiration in plants. The TCA cycle starts the condensation of the acetyl group with oxaloacetic acid. This is a direct question. Option number 3 is the correct answer. Next one, this is coming from plant growth and development, where you need to match the plant growth hormones and the compound from which they are derived. So let's look. Now auxin is mapped or is derived from indole acetic compound. D should be going to 2. So directly I can eliminate options 2 and 3. Then kinetin is derived from adenine. So C has to go to 1. So C is going to 1 in the first option, and I am at the correct answer for the question. Next one, when pollen grains of a flower of a plant pollinate the stigma of the flower of another plant. Let us understand this by a diagram. Now, if the pollen grain is going to pollinate the same flower, I will call it as self-pollination or autogamy. Now, if the pollen grain is going to pollinate another flower on the same plant, here I will call it G-itenogamy. Now, if the pollen grain of one flower on one plant is to pollinate another flower on another plant, this is what I will call as xenogamy or cross pollination. So hence, by concept, the correct answer for this question is going to be option number 3, that is xenogamy. Next one, fusion of a male gamete with the central cell in the embryo sac of an angiosperm is called triple fusion, because there is one male gamete and in the central cell we are going to have 2 polar nuclei. The fusion of 3 haploid structures is what is called as triple fusion. Hence, for this question, option number 4 is the right answer. Next, now here they are given us the structure of the pollen grain. Now, before reading such an elaborate question, it's always advised to understand the diagram. So, A is going to be the vegetative cell, which has the vegetative nucleus and give rise to the pollen tube. B is going to be the generative cell that is going to give rise to 2 male gametes, and it is going to float in the cytoplasm of the vegetative cell. So once we know that, with that we are going to get option number 2 to be the right answer for this question. Next, how many types of gametes will be formed by a parent with genotype capital A small a, capital B small b, capital C small c. Now to this we have a formula, that is 2 to the power n, where n is number of heterozygotes. So in this case, capital A small a, capital B small b, capital C small c, n is equal to 3. Therefore, number of gametes is equals to 2 to the power 3, which is equal to 8. Therefore, option number 1 is going to be the correct answer. Next, when a single gene exhibits multiple phenotypic expression, the phenomenon is called as pleiotropy. Now this you can recollect, there is a gene B or small b. Now here they are going to control the size of the P and also the shape of the P grain. So a single gene is controlling two features, that is the definition of pleiotropy. Next, a color blind man marries a carrier woman. So here we are talking about X-linked recessive disorder, that is color blindness. Now man cross with woman. Man is color blind, so X, CB, Y. Then the woman is carrier. Now here you have to pay attention. Females have two X chromosomes. If one is defective, we call them as carrier. If both are defective, we call them as affected females. So in this case, this one. Now these are the gametes. Now we are going to get XXCB. This is carrier female. This is affected or color blind female, XY. This is normal male, and then I have XCBY. This is color blind male. Now they are asking the percentage of their color blind progeny. So percentage of color blind progeny is 2 divided by total number of progeny into 100, which is 50. So therefore, for this question, answer is option number 4. Next, here they are presenting a question which is asking you about the chromosomal disorders. So the Down syndrome and the Turner syndrome. So Down syndrome, you should understand, it is a trisomy of chromosome number 21. 3 copies of the 21st chromosome. Turner syndrome is observed in males where there is a deletion of the X chromosome. Since it is an autosomal disorder, it affects males and females equally. Now let us look at the options. Down syndrome, short statured individual is true. Gynecomastia in men is false, because this is a affected pattern that we see in Klinefelter syndrome. So therefore option A is false. Round head, partially open mouth is true. Overall masculine development is false. We don't see too much of a masculine development in Turner syndrome. Now so therefore B is false. C. Broad palm, physical and mental development retarded is true. Sterile females with rudimentary ovaries. So therefore option number 1 is the right answer for this question. Next, RNA polymerase. RNA polymerase 2 is responsible for the transcription of hnRNA, option number 1. This is the primary transcript of the mRNA in a eukaryotic cell. Next, which of the following enzymes increase the permeability of the bacterial cell to lactose? So this is the question on lac operon concept. So here we will have LAC-Z gene, which codes for the enzyme beta galactosidase, which helps in cleaving the lactose into simple sugars. Then we have the LAC-Y gene, that is called beta galactosidase permease, which is the actual enzyme which we are talking about for this question, that increases the permeability of the cell to lactose. So the answer is option number 4, beta galactosidase permease enzyme encoded by the LAC-Y gene. The third gene is LAC-A gene, which is called beta galactosidase transacetylase gene. Next one, which of the following statements are correct with reference to prokaryotic genome? So with respect to the haploid set, what is the correct set of statements for the prokaryote like bacteria? Monocystonic structural gene is false. because it's a characteristic of eukaryote. Prokaryote, we are going to see polycistronic genes. Introns are absent, true. That's why they don't need processing. Transcription and translation are coupled processes, true, because both of them are going to happen in the same chamber and translation is going to begin even before the completion of transcription. Primary transcript undergoes splicing is a false statement for prokaryote, seen only in eukaryote. Only one RNA polymerase is present for a prokaryote, that is true. So therefore, we have got B, C and E. So therefore, option number 4 is going to be the correct statement for this question. Next, the drug prescribed to the patient who undergone organ transplant is cyclosporin A and is produced by the fungi Trichoderma polysporum. This is a direct question from Microbes in Human Welfare chapter. Next, the following statement, again another question from Microbes chapter. Statement 1, biocontrol. So biocontrol is a method of controlling or keeping the pest under control, not completely eradicating them by the use of other biological organisms. Biocontrol refers to use of biological methods for controlling plant diseases and pests. So therefore, statement 1 is true. Statement 2, Trichoderma species are effective biocontrol agents for several plant pathogens. This is also a true statement. Therefore, we can say that both statement 1 and 2 are correct. Hence, option 2 is going to be the correct option for this question. Now, we are coming to another question which has matched the following type. So, column 1 you have the names of some organisms and column 2 their utility. So, Streptococcus is a bacteria from which we can obtain the clot buster enzyme, that is streptokinase. So, A going to 2. I can directly remove options 1 and 2. Then penicillium is the organism which gives us the antibiotic penicillin. So, penicillium is a source of antibiotic, B going to 3. So, directly option number 3 is your correct answer for this question. Now, here is a question which says a scientist wants to produce virus-free plant in tissue culture. Which part of the plant will he use as an explant? So, ideally we would remember that meristems are those cells of the plants that are in a state of continuous and active cell division. Now, once a virus infects the cell, even before it can synthesize its components, the meristem has undergone several divisions. So, therefore, meristem is the best candidate to choose and create plants that are free from viral infections. So, from the given options, axillary meristem and apical meristem both are going to qualify. Hence, option 4 is going to be the correct answer for this question. Next, which one of the following population attributes contributes to the increase in population density? We are aware of 4 population attributes. Natality is birth, mortality is death, immigration is coming into a place, emigration is moving out of the place. So, based on these concepts, it is evident that natality and immigration are those 2 attributes that are going to contribute to increase in population density. Next one. Now, this is a question from the chapter Organisms and Population. It says that 8 individuals in a laboratory population of 80 fruit flies died during a specified time interval. Now, we have to calculate what is the death rate. So, here the death rate is given by the simple formula, that is number of organisms died divided by total number of organisms. So, number of organisms died is 80, total number of organisms died is 8, total number of organisms is 80. So, when we simplify this, 1 by 10 or 0.1 individuals per time interval. So, therefore, option number 1 is going to be the correct answer for this question. Next, This is the question from ecosystem chapter, which says what is the correct sequence of steps involved in decomposition. So the first one is going to be fragmentation, where the larger portion of the detritus is made into smaller pieces. Then is leaching, wherein the inorganic nutrients are going to be precipitated as unavailable salts. Then catabolism is further breakdown, or we call as destructive metabolism. Then the next step is humification, that is formation of a dark colored substance which is rich in organic nutrients, and the last step is going to be mineralization. So this is the correct order. So if we have to look for this, option 2 is going to give us the correct answer for this question. So now I would like to call upon my colleague Mr. Zamir Ahmed to please continue the live discussion of the biology paper. Thank you so much.
Thank you, sir.
Thank you, sir.
It was a wonderful discussion, and I hope all your doubts, all your queries regarding the botany paper are solved. As sir introduced, my name is Zamir, and from here on I will be handling the zoology part of your KCET. Right? The first question in the zoology part: during a field trip, a student observed a marine organism. So this is the first character which they observed, right? So you can think of any organism which is exclusively marine or it is marine with a worm-like body. So they are saying it is a worm, which is cylindrical, and body was divisible into proboscis, collar and a long trunk. Now this is a characteristic feature of a phylum which is called as Hemichordata, isn't it? The primary example in the NCERT image which is given for Hemichordata is of Balanoglossus. So this is proboscis, this is collar, and this will be the trunk, isn't it? So the answer is Balanoglossus. Pterophyllum, trigon are examples or Pisces, and Ophiuria is an example of an echinoderm. So we can move ahead to the next question. Which of the following statements are correct, they are asking, with respect to frogs, right? They are saying Bidder's canals are present in case of male frogs. I agree to it, but let us hold on to this because I am going to explain this to you in the next slide. Copulatory pads are present in case of female frogs, which is an incorrect statement, because copulatory pads are also a characteristic feature of sexual dimorphism in case of frogs, wherein they are present on the first digit of the forelimb of male frogs. It is not female frogs. Let us move on. Sound producing vocal sacs are present in case of male frogs. This is also true, which will help them in croaking, which is a mating call in case of male frogs. Last but not least, cloaca is present in male frog only. Incorrect. Cloaca is a common structure which has the openings for alimentary canal, excretory system, as well as the reproductive system, which is present in both males as well as female frogs. Now let us understand this structure which we spoke about, Bidder's canal. I told it is present only in males. How? This part of the image, let us understand, this is in case of males, this is in case of females. We are going to talk about the reproductive and the urinary system in case of frogs. Imagine if this is the alimentary canal, it opens up into this large sac called as cloaca, and this aperture is called as the cloacal aperture, right? Let's talk about males first. This will be the kidney of the frog, and on that you can see presence of adrenal gland, right? We also have testis here. Above the testis you can see presence of the fat bodies. Will not get into much details. Now the seminiferous tubules, the vasa efferentia are bringing in the sperms into the urinary system of the male frog. Here we have a canal, this canal where the vasa efferentia is opening, this is called as the Bidder's canal, through which it will open into the transverse canals, longitudinal canals, eventually into the ureter. That is why this can be called as urinogenital duct in case of male frogs, because it carries both sperms as well as urine. Whereas in case of female frogs, you can see this is the kidney, This is the adrenal gland. These are the ovaries, right? But they do not have any anatomical relationship between each other. There are no tubes which are passing. So here the oviducts collect the eggs and directly pour it into the cloacal chamber. So Bidder's canal is only present in case of frog. Now you might agree to this. Out of this what they have asked, how many of them are correct? So A is correct and C is correct. That will give us option 1. Let us move ahead. The graph showing the concept of activation energy in the activation energy of an enzyme is given below. Observe the graph and choose the correct option for M and N. This is a very relevant graph. All of you have seen this in biomolecules. We already understand the definition of activation energy. This is the minimum amount of energy which is needed to initiate any biological reaction in biological terms, isn't it? We also know presence of enzymes will lower the activation energy. So in this graph, less amount of activation energy is needed in this, correct? So here enzymes are present, whereas in case of M, the activation energy is more. So you can say enzymes are absent, which will give us M activation energy without an enzyme, N is the activation energy with the enzyme, that is option 3. Moving ahead, the respiratory mechanism controlled by medulla oblongata, so they are saying controlled by medulla oblongata can be altered by, who can alter that? They are saying both pneumotaxic and chemosensitive areas of pons and medulla oblongata. Here, this is a little confusing, but you have to understand the fact that the pneumotaxic center is present in pons, right? The respiratory system along with the chemosensitive areas is present in the medulla oblongata. Together, both the pneumotaxic center of pons and chemosensitive areas of medulla can control the respiratory mechanism. So that will give us option 1. Next, which among the 3 layers of blood vessels, tunica intima, tunica media, tunica externa, is comparatively thin in the veins, they are asking. For this you have to understand 3 things. 1, artery, vein, and another thing, capillary, right? So in the artery, if you take a transverse section, all these 3 layers can be visible. In veins, when you take a transverse section, all these 3 layers can be visible. But there is a very prominent difference between the arteries and veins. 3 layers they told. Tunica intima is basically a thin layer of squamous epithelium, right? Here also tunica intima is there. Is there a difference? No. Endothelial cells, squamous epithelial cells. Tunica media is mainly made up of smooth muscle cells. Is mainly made up of smooth muscle cells present in both arteries as well as veins. But comparatively, where is it thicker? It is thicker in case of arteries. You can also see the lumen of the arteries is narrower when you compare to lumen of the veins. Valves will be present in the veins, whereas valves will be absent in the artery. Capillary is only made up of tunica interna or tunica intima, that is squamous cells. That is only the squamous epithelial cells. So the major difference is here in the tunica media part. Tunica externa is a thick layer of connective tissue, almost similar in case of arteries as well as veins. So the major difference lies where? In tunica media. Ignore the jumbling of the options, but remember the answer is tunica media. Next, in nephron, transport of substances like sodium chloride and urea is facilitated by special arrangement called as counter current mechanism that comprises of. Now you can focus on 2 things. 1, you can get into the route of sodium chloride, right? Water, urea, where is it permeable, where it is not. Or you can focus on a small phrase here which says called counter current mechanism that comprises of. They are just asking you what does the counter current mechanism comprise of. Generally a nephron has these parts: Bowman's capsule, PCT, descending limb of Henle's loop, ascending limb of Henle's loop, DCT, and collecting duct. Whereas around the Henle's loop, you can clearly see presence of an intricate network of capillaries, In this case, why is this called as countercurrent mechanism? Because the filtrate is flowing downwards here towards the medulla, whereas here it is flowing upwards. Whereas the blood in the vasa recta is flowing downwards here and upwards here. So the blood and the filtrate are in different directions, that is counter current. That is why it is called as counter current mechanism. And two structures which are primarily involved here is Henle's loop and vasa recta. So this is an easier way to arrive at the conclusion for this question. Alright, next question. In the mechanism of muscle contraction or shortening of the muscles, the dash gets reduced whereas the dash retain their length. For this, we have to understand the ultrastructure of muscles. A very simple diagram we will draw, right? Let's assume that this is the condition when it is uncontracted, right? I hope all of you know this. I don't want to brush your memory right now, correct? So this part of the sarcomere is called as the A band, dark band. Both thin and thick filaments are there. This part of the sarcomere is called as the I band, right? This part of the sarcomere is called as the H zone. And the hypothetical middle line is called as the M line. Now what they are asking, what gets reduced and what remains the same? Now whenever the myosin heads will bind onto the actin filament and perform something called as a power stroke, the thin filaments of the sarcomere are pulled towards the M line, So post contraction, what we can see in a sarcomere is that what has vanished? Light bands have vanished or reduced, right? What remains the same? A band. So the dark bands has remained same in length, whereas you can say this has reduced or this has vanished completely. So that is why you can say I bands get reduced, whereas A bands will remain same. Option 4 will be your answer. Next, identify the correct sequence of action potential as it arrives at the axon terminal from the given choices below. Now let us understand this. Telodendria, axon terminal forms this presynaptic knob. Inside the presynaptic knobs, what are present? Synaptic vesicles are present. Inside the synaptic vesicles, what is there? Neurotransmitters are there. As and when action potential arrives at the presynaptic knob through the axon terminal, here what happens is through exocytosis, the neurotransmitters are released into a space called as the synaptic cleft. Here what is there? Assume that this is the postsynaptic neuron. Generally assume that this is a dendrite of a postsynaptic neuron, right? So these neurotransmitters will bind onto the receptors, specific receptors present on the postsynaptic neuron. Here what will happen? Again they will open up the channels. Here action potential is transmitted. So the sequence should be axon terminal, it will come to the synaptic vesicles. Through that it will come into the synaptic cleft, the neurotransmitters, which will go and bind on to the postsynaptic membrane and will be transmitted into the postsynaptic neuron. If you see, option 3 basically follows whatever we have told here in this case. So right option is option 3. Next, identify the statements given below that does not correspond to the functions of cortisol. Now we know for a fact that adrenal gland has adrenal medulla and adrenal cortex, correct? Now adrenal gland secretes a group of hormones called as glucocorticoids, out of which cortisol is one, and clearly NCERT mentions that it helps in maintenance of cardiovascular system and also involves in kidney functions. It produces anti-inflammatory responses or reactions, because of which suppression of immune response will be there, right? It also stimulates in RBC production, but something which it does not do is option 3, that is maintains electrolytic balance, osmosis and blood pressure. This is the function of aldosterone, not cortisol. So you can say option 1, that is 3 only. Next, match the hormone with its site of production. First one, HCG and HPL. This is human chorionic gonadotropin, chorion, right? Human placental lactogen, placenta. This is also called as human chorionic somatomamotropin these days. So basically, these are released at the placenta. So you can say A is 2. Progesterone. Progesterone is secreted by the corpus luteum. Large amounts of progesterone, some amount of estrogen helps in maintenance of the endometrium. Androgen, male sex hormones, primarily testosterone secreted by Leydig cells. Relaxin is a hormone secreted by the ovary, helps in expansion of the pubic symphysis, helps in parturition later. So A is 2, B is 3, C is 4 and D is 1, that is option 2. This will be the right option for this case. Next, choose the correct sequence of sperm transport during ejaculation. For this, we will need a LS of testis. A brief explanation, please follow this. Here we have the lateral section of testis. Testis is divided into, about 100 to 150 lobes, in which about 1, 2, 3 seminiferous tubules will be present. Here is where spermatogenesis will occur. From here, it will enter into a labyrinth network of tubules called as retetestis. First is seminiferous tubule, second is retetestis. From here, it will enter into vasa efferentia. So third region is vasa efferentia. From here, it will enter into a inverted comma shaped structure. When coiled looks very short, but when uncoiled 6 meters. So this will be the fourth region, epididymis. Last will be the vas deferens. This will be the vas deferens. Now, another diagram, this will be the urinary bladder, right? Behind the urinary bladder, vas deferens is coming. Along with this, you can see the opening of seminal vesicles. Together, they form a structure called as ejaculatory duct, right? After which it enters into the urethra. So, the correct sequence would be seminiferous tubules, retetestis, vasa efferentia, epididymis, vas deferens, and then ejaculatory duct, and then urethra. Through the urethral meatus, it will go out during ejaculation. Option 3 is your right option. Next, select the mismatched pair. This is during the development of fetus, gestation. First month of pregnancy, they are saying formation of heart. Correct. First signs that the embryo is alive and can be heard through a stethoscope. Second month of pregnancy, they are saying movement of fetus. No, movement of fetus generally occurs during the 5th month of pregnancy. It occurs during the 5th month of pregnancy. Third month of pregnancy, formation of most of the major organ system, right? Like hands, limbs, genital organs would be developed. 6th month of pregnancy, eyelids separate, eyelashes are formed, even body hair will be developed at this point of time. So A is right, B is incorrect, C is right and D is right. And they are asking the mismatched pair, that is B. So option 4 is the right answer. Out of the following options, identify which is not a natural method of contraception. Very easy. Answer is implants, because they are chemical methods. Here in this case, you can see lactational amenorrhea, temporary phase for 6 months. No pregnancy can occur because of inhibitory effects of prolactin. Periodic abstinence. Here the people will abstain from intercourse during the unsafe period, where the probability of fertilization is more. Coitus interruptus. During copulation, the male will withdraw the penis right before the ejaculation. Out of this, the failure rates are very high, but the question is not that. Which one is not a natural method of contraception? Answer is implants. Let's move on to the next question. In zygote intrafallopian tube transfer, so that is ZIFT, right? The embryo up to fallopian tube is transferred into the. Now, we have to understand, during assisted reproductive technologies, in vitro fertilization, what happens? If it is before or at 8 cell stage, 8 blastomeres, it can be transferred directly into the fallopian tube. That is why it is called as zygote intrafallopian tube transfer. If it is after 8 cell stage, more than 8 cell stage, it will be transferred into the uterus and will be called as intrauterine transfer. Above 8 blastomeres, it will be transferred into the uterus. So the answer here is 8 blastomeres, option 1. Read the following statements. I made a primary mistake while reading the question, so it is logical, even you would have. MTP is to, both are NCERT statements, one word is wrong here. MTP is to get rid of wanted pregnancies. This should have been unwanted pregnancies due to casual unprotected intercourse or failure of contraceptives used during coitus or rapes. Obviously, statement 1 is incorrect. Statement 2, MTPs are performed legally by qualified doctors giving proper medical justification. This is obviously right. So, statement 1 is incorrect and statement 2 is correct. Option 2 is the right option. When a change in the gene frequency of population occurs by chance, it is called, so random change in the allelic frequency of a population is called as genetic drift. That is the actual definition of genetic drift. Genetic recombination generally occurs during meiosis, where crossing over of chromosomes genetic material occurs. Founder effect is some part of the population will move out, and in the new population the allelic frequency is completely different. So in that population, the original people who moved there, this is called as basically the founder's effect. Gene migration can be basically addition of alleles or deletion of alleles from a certain population due to immigration or emigration, can be called as gene migration. Darwin's finches represent one of the best examples of, basically we use this term or Darwin's finches to basically talk about adaptive radiation. It is not chemical evolution. It occurred when there was no oxygen in a reducing atmosphere. Genetic equilibrium, no. Seasonal migration, no. What did Darwin's finch do? Original seed-eating birds migrated into different islands where there were no seeds. Imagine if there were worms. Their beak shape changed to accommodate them to feed better on worms or any other material which is available there, due to which speciation occurred. So this is called as adaptive radiation. Next, choose the correct statements from the following. Charles Darwin traveled around the world in a ship called as Her Majesty's Service Beagle. Yes, HMS Beagle, that is true. There has been gradual evolution of life forms. This is what Darwin says, true. According to Darwin, fitness refers to physical fitness. This is incorrect because he refers to reproductive fitness whenever he is talking about fitness. Fossils are the remains of hard parts of life forms found in rocks. True statement. Hugo de Vries, this should be Hugo de Vries, a naturalist, worked in Malay archipelago. Incorrect statement. Hugo de Vries worked on Inithera lamarckina, who in turn gave the mutation theory. Alfred Wallace was the person who worked in Malay archipelago. So A is right, B is right, and then we have D. So A, B and D are correct. Option 1 will be your right answer. Which of the following HIV replicates and produces progeny viruses? Again, you will get confused in this point. Two things can happen. Once the HIV virus infects a human, primarily it enters into macrophages, wherein under the action of reverse transcriptase, what it does? It produces viral DNA. It does not produce RNA, right? They're talking about progeny virus. So that happens in T helper cells. Eventually, they'll reduce in number, because of which the immunity will be less. That is why it is called as acquired immunodeficiency syndrome, isn't it? So the answer should be helper T lymphocytes. The progeny viruses are produced in helper T lymphocytes, Th cells. Which of the following are techniques for detection of cancer of internal organs? NCERT statement you can pick up. Radiography example is given, X-ray, right? This is true. MRI is also used. Magnetic resonance imaging. MRI is used. CT scans are used. Computed tomography. Vidal test. This is a typhoid test. Incorrect. Vidal test. Typhoid test. Incorrect. So A and B. That will be our option C. Malignant malaria is caused by direct statement again from NCERT. The answer is Plasmodium falciparum. It says this is one of the most serious types of malaria and can be fatal as well. Next, match the following. This is from applications of biotechnology. Bioreactors are basically used, or they can say they are the vessels used to convert raw materials into specific product. Downstream processing involves separation and purification of one of the last stages. Recombinant protein, you can think of insulin produced by rDNA technology. PCR, polymerase chain reaction, detect mutated genes in susceptible cancer patients. Directly it does not, helps in amplification of DNA. Through other methods we detect cancer causing sequences in that DNA, right? So A is option 2, B is option 4, so that would give us option 3. A is 2, B is 4, C is 1 and D is 3. The part of plasmid that codes for proteins involved in replication of PBR322. Basically, this is where we tend to lose marks. In NCERT, if you see, just beneath the diagram of PBR322, there is a yellow colored box which we tend to ignore. That is where this statement is mentioned and says ROP is the region which basically codes for the proteins involved in replication of PBR322 plasmid. So this tells us we should be conscious about everything which is in NCERT. Next, to isolate the DNA from fungal cells, bacterial cells, plant cells, the enzymes required are respectively. Primarily we need what? We need to break through their cell wall of all these three. Cell wall of fungi, chitin, so chitinase, bacteria, lysozyme and plants cellulase. So this is option 1. Option 1 is the right answer. Mature insulin, which immature insulin, which of the peptide is not present? So basically immature insulin or the proinsulin, here we have the A and let's think this is B, right, and this is C. If you remove the C chain of proinsulin, we will be left with A and B chains, which is called as the mature insulin. So this is C peptide. Some strains of Bacillus thuringiensis produce proteins that kill insects. Remember this word, insects. This will help you arrive at the answer. Which one of the following is not killed by proteins? They kill insects of Lepidopterans, Coleopterans, Dipterans, but they don't kill tapeworms, isn't it? They are platyhelminths. So option 2 is the right answer, right? So that brings us to the end of the session. I hope all of you would have enjoyed and learned and analyzed the papers of all the KCET exams which happened this year. So let's meet during the NEET examination. I wish you all the best for KCET and all the other competitive examinations you are taking up. Thank you so much.